tonight we have the distinct pleasure of hearing Dr. Joel Mercado Diaz, and he is a lichenologist and plant biologist with academic training in environmental science and ecology systematics. His research centers on the phylogenetic systematics and ecology of lichens and plants in Puerto Rico and the Caribbean, with additional interest in natural history collections, the history of botany and lichenology, and forest conservation. He earned a PhD in evolutionary biology from the University of Chicago in 2021 through the Committee on Evolutionary Biology and was a resident graduate student at the Field Museum under the mentorship of Dr. H. Thornstern Lombosch. I'm hoping I didn't murder that. His work integrates taxonomic, ecological, and comparative phylogenetic approaches to study lichen diversity and distribution across the Caribbean islands. He currently works as an independent contractor, providing scientific writing, taxonomic inventories, and species identification services. But really, the best feature that I enjoy the most often about Dr. Joel is his curation of iNaturalist observations on lichens in Puerto Rico. And he has produced a guide to the Puerto Rican lichens in El Yunque that I absolutely adore. And I will do my best to link in the chat while he presents to you all. So without further ado and with great pleasure, I leave you with Dr. Joel Diaz.
Hello, everyone. Good evening to everyone. Thanks for that amazing introduction, Serenella. I'm already having a lot of fun hearing you guys. Thanks for all those words. Hi from Puerto Rico. I'm in Guaynabo, Puerto Rico, right now. Um, I actually wanna. I just realized that I'm not the only person from the island joining. So here's like some friends, Miriam Toro, Kurt Miller, which you probably have heard before in some of your talks. Kurt, hi, and Doralis is around. So there's some other friends, but anyway, I just want to take the time to, say hi to them too, and hi to you guys. So I'm gonna let's see if I get this is still working. Let's do a share. Give me just a sec until I get set. Just a second. Let me know if you're seeing what I'm seeing here. You get it? Do you hear me? You see me?
Looks good.
Good to go? Okay, guys. Thank you for joining to hear a little bit of some of my boring stories regarding lichens of the Caribbean. So let's just get started. Let's see. Oop, it's not. Huh. Are you getting my slides? Having difficulties. Oh, there you go. Okay. So I'm gonna start my talk dedicating it to the memory of Dr. Ismael Landron Concepcion. You probably haven't heard this name ever. So Dr. Ismael Landron was my early mentor. We lost him in 2022. He's actually the first native lichenologist, first native Caribbean lichenologist. He's from Puerto Rico. He obtained his PhD from Michigan State University, and he did really cool taxonomic work on the lichen genus Ramalina, which you have all over the continental US. So before that, and in the memory of him, I want to dedicate this talk to him. I just also wanted you guys to meet him and know about him. So let's start. I want to start talking about islands. So I don't need to convince you. Islands are amazing ecosystems, right? They don't cover a lot of surface area in the planet. Actually, they just cover 3.5% of the surface area, but their contribution to biodiversity is astounding. It's 15% of all the species that we have, and that is the reason why many scientists consider islands, from an evolutionary biology perspective and ecology, as amazing natural laboratories for understanding patterns and processes in evolution and ecology. So before getting into lichens, I wanted to start just giving a geographic context of the area we're going to be covering today. And this is the insular Caribbean region. I make the distinction because they're Caribbean parts of the continental America or the neotropics. But I'm going to be focusing on the islands. There are three main aspects of this island that I consider amazing and what makes them special. First, they're incredibly heterogeneous environmentally. So we have elfin woodlands in high elevation. We have pine forest, native pine forest, and that goes all the way to the coast to really warm and humid wetlands and mangrove ecosystems. They're they're an assembly of really complex islands in terms of the geology. So we have the Greater Antilles, which are comprised by Cuba, Jamaica, Hispaniola, and Puerto Rico. These are really, really old islands, have more or less their history goes back to 130 million years ago. They're just basically fragments of continental crust that ended up there. We also have the Lesser Antilles, which is the easternmost islands. These are or of more recent geologic history. These are volcanic of recent volcanic origin, and as you know, we have the Bahama Islands. These are even of more recent geologic history, more associated with recent sedimentary processes in that part of the planet. Another amazing aspect of the islands, which makes them unique to, let's say, Hawaii or Galapagos, and is that they are sort of like intermittently connected to the continents. That means that they are isolated enough, but close enough to continental land masses to have some shared species, but also allow evolution to play and have endemics, for example. So that those are three main aspects that makes these islands really special for evolution. They have contributed a lot to our understanding of evolutionary and ecological processes. The most fascinating aspect that have been studied is probably the case of adaptive radiations in the anolis lizards. This is an amazing example of replicated evolution of similar morphologies that have evolved independently in each of these islands in this group of organisms. So this type of studies led by, for example, Jonathan Lossos and other colleagues have allowed us to understand important concepts such as contingency and determinism in evolution. So again, just building up, making the case that this is not just a regular set of islands. This is a really special set of islands. So hopefully I don't need to, I already talked a little bit about the islands. I want to delve into the lichens. You're a mycological association. I was hoping I don't have to explain a lot about what lichens are, but it's just worth it. So lichens are a symbiotic association, as most of you know. These are fungi that associate with algae and or cyanobacteria, and also with a really complex set of microorganisms, a microbiome. So the relationship of these organisms make this community of species that are lichens. So there are many growth forms, as you already know and already saw from all the pictures shared by Natalie and the rest. But the three main ones would be crustose growth forms. These are lichens that grow embedded to the substrates. We have foliose lichens. These are the leafy looking lichens growing in their native substrates. And then the fruticose lichens. These are lichens that have more of like a 3D appearance. They tend to look shrubby or like pendant. So those are the main visual cues to recognize lichens. They have these appearances. So, as I already said, this is a symbiotic association. So the key, I wanted to bring this slide because of this, especially the species you're seeing here, which most of you are familiarized, Santoria parietina. I wanted to say here that Santoria parietina is the name of a fungus, correct? So we name lichens following the fungal nomenclature. The algal or cyanobacterial species living within have their own nomenclature and taxonomy. So the picture above to the right is Santoria parietina in culture. So as you see, when you grow it without its symbiotic partner, it grows completely different. There's another example of Acarospora fuscata to the lower right. they're two symbionts growing separately in vitro. So I wanted to make that distinction, and also just letting you know that there are different numbers and estimates of lichens, but one of the most recent ones is 20,000 species of lichens. This is an estimate by Robert Lukin of 2017. These organisms, if you have seen, are amazing in terms of their variation in color and shapes, and the lichens in the Caribbean are no exception to that. So let's go into it. We can argue a lot about when lichenology in the Caribbean started. I'm gonna make the case that it probably started in 1696 with the work of Hans Sloane. This is an Irish botanist that did taxonomic work mostly on Jamaica. He had two publications from that year in 1707. Almost 100 years later, the Swedish scientist Olof Swartz published his famous Prodromus. This is still a really relevant piece of work from Swartz, as many of the names of lichens, of mostly pantropical lichens, are still valid. A lot of the epithets are still used, and most of them are curiously considered to be widespread, but some of our work is starting to reveal that some of these names actually correspond to Caribbean endemics. So I'm gonna be talking a little bit more about that later, but just wanted to give you a heads up about that. So a little bit later, we have mostly works in most of the work. The early work was done in Cuba by North American scientists. Charles Wright made a lot of collections in Cuba. Those were mostly identified by his colleague Edward Tuckerman, and the actually, a lot of the specimens were sent to the lichenologist William Nylander, who identified a lot of the Cuban material. There was a lot of Cuban, but also Puerto Rican material processed and identified by the lichenologist. The Swiss, I think it was from Switzerland, Jan Muller Argeviensis. And this is actually to the right of the first formal publication of Puerto Rican lichens that dates to the 1898, if I remember correctly. So we have been looking at lichens in this region for a while. However, and there have been many people throughout the history have done multiple works. I just wanted to jump into them to people that are doing current work. So today, Caribbean lichenology, albeit a little bit slow, is still active. I just want to mention Dania Rosabal. So Dania is a Cuban lichenologist, and her work is really cool. She has published few papers focusing mostly on ecological aspects of lichens in the forests, in the different type of forests in Cuba. More recent work by our French colleague Elise Le Breton. She's a French PhD student in Belgium, has done astounding work combining different types of data, from molecular data to species distribution data, historical collections, to shed light on evolutionary and biogeographical aspects of folliculous lichens. These are lichens that grow on leaves, and her work focuses mostly. It goes all over the place, but she has done a lot of work in Guadeloupe. So she's doing great work advancing knowledge in this region. And of course, I have to mention Robert Luking from Germany and Viviana Moncada from Colombia. They are involved right now in a really massive project looking at lichens in Cuba for the the ongoing Flora of Cuba project. They are doing a lot of stuff, updating checklists, but they have published also really cool stuff, such as this new genus called Saxiloba, this enigmatic genus with a really unique morphology within the family Porinaceae. So these are the main players. We have many others, such as Andrea Uproot, Harry Sitman, that have worked in this region. But these are, actually people that are focusing on more aspects, on aspects that go beyond taxonomy and species identification that I wanted to highlight. Anyway, so how many lichens are there? I don't have a straight answer to that, because as you will see, there's not many lichenologists around. But there are studies, and this is one from 2009, from Robert actually, and some other colleagues, that they tried to use something called the species area relationship, um, to try to estimate the number of species for different countries in the neotropics. So I extracted from that paper the data that belong to the different islands of the Caribbean. So in this figure, you're seeing this as a two y-axis. To the left, we have the surface area in square kilometers, and the y-axis to the right is the estimated number of species. And as you can see, and as would be expected, and we have in the x-axis, of course, the different islands that from the Caribbean. And as is expected, we see more species in the islands that are bigger, such as Cuba, and less species in the smaller islands, such as Guadeloupe and Dominica. But something that really blows my mind from these numbers is what you see in Guadeloupe and Dominica, right? We have a lot of species that are estimated, uh, predicted to occur in really small surface area, and that's something distinctive of the tropical rainforest, correct? That predominate in these islands. I did a similar exercise, that basically I applied the same idea of Robert using the species area relationship to estimate number of species based on the size of an island. But here I embedded I tried to do, before doing that, I went to the literature and extracted the different species that are known from the literature for each of these islands. So to the left, we have number of actual species in the literature. This was done nearly some years ago, so this has changed, of course, but it gives us an idea. So we have to the left the number of species in the literature and their area in the x-axis. And as you can see, and something that it's easy to see, is the largest islands, Cuba and Hispaniola. These islands have a low species richness in the literature, but it has a high expected number of species based on this simple species area relationship. I wanted to point out that I used, at the point of doing this graph, I knew a lot about Puerto Rican lichens, so I used the number of species of Puerto Rico to calibrate my curve. So that's how I got that. But again, it's similar to the previous one, and it tells and makes the case that these are highly diverse islands that we know very little of, right? Based on the numbers and the data. So how many species in the Insular Caribbean? So that question, to answer that, we also have to mention the work of Dr. Henry Imshog. He published the Catalog of West Indian Lichens in 1957, and I did the tally of the number of the species there, and he that sums to 1,700 species. But once again, based on those estimates, we see, for example, in this previous graph, from Robert, that, for example, for Cuba, you have nearly 2,000 species. So just for one island, an estimated number of species of 2,000 is quite high. So we know that this number is really probably low. But the work we have, the very limited work inventory work we have for these islands, tells us that, for example, in Cuba there are one ongoing and a previous work documenting between 1,100 to 1,027 species. In Puerto Rico, 1,000 to 100. Sorry, 1,100, I should say. So as you see, these are numbers that I consider low based on the previous predicted number of species for these islands. But once again, this is just based on the literature. You need to consider that there's a lot of synonymy, like a lot of names are heterotypic synonyms. So they're probably talking about the same species, but one species can have multiple names in this checklist and inventories. So again, I'm sorry I don't have a formal strict number, but it's probably between those 1,500 to 3,000 species in this region. So I began working with lichens more formally, doing morphology based work, maybe early late 2000s. So this is one of my first studies focusing on the genus Cynogonium. In this study from 2013, we described 4 new species of Cynogonium from what we consider vulnerable ecosystems in Puerto Rico, and we make the case that these 4 species are potentially threatened in the island. And I wanted to mention this publication we got out in 2014. This is again based on traditional taxonomy, morphology-based taxonomy, where we describe 2 new genera and 12 new species of lichens in the family Graphidaceae that I collected in El Yunque National Forest here in Puerto Rico. And this is probably the first paper and study that makes the argument that there's probably hiding the high diversity hiding in this undersampled ecosystems in the Caribbean, especially Puerto Rico, and potentially higher endemism that it's waiting to be discovered. So the game changed for me when I in 2015 when I moved to Chicago to do my PhD, and there I was able to apply integrative taxonomy, which is the what I did in the previous studies, combining morphological data, ecological data to understand our diversity. But this time I was able to incorporate the genetic component to it. So to go on, I wanted to mention that one of the first things I did is focusing on a genus of lichens in Puerto Rico, and I decided to go with the genus Sticta. This is a foliose group of lichens that were really poorly understood in Puerto Rico. So to give you an idea, we knew about Sticta that there were at least 9 morpho species that were documented by Dick Harris from the New York Botanical Gardens. And I'm saying morpho species because they didn't even have a name. So we knew from their morphology that they were 9 different from that early work in 1989 from Dick Harris. But I revisited this genus, this time doing intensive sampling throughout different ecosystems in Puerto Rico and incorporating molecular markers and genetic data and phylogenetic analysis. And that work allowed me to reveal that there were at least 16 species, 11 of these potentially endemic to the island of Puerto Rico. We also uncover very low cryptic diversity. And that means that, most of the species are easy to recognize just based on morphology, and that a lot of the communities are basically polyphyletic. That means that they probably emerged from independent colonization events throughout evolutionary time. And the most interesting fact I think that I found is that most of the lineages from Puerto Rico had a tendency to be associated with lineages from South America. So this quick view to this paper, I wanted to mention because it was an inspiration to the 2 case studies a little bit of that I wanted to share with you some information. So, and to tell you more about that story of the diversity and evolution of lichens in this region. So the first one, I'm gonna focus on the same genus, Sticta, but this time looking at species all over the islands of the Caribbean. And this is work that I did at the species level. You see, I'm gonna focus on another study that goes into more population level details. But this first part is gonna focus on more like intra-island comparisons and species level information. So for this 2023 study, the guiding question was what were the main ecological and evolutionary factors shaping the diversity of Sticta in this region? And this study had 2 components, a macroevolutionary component and a community phylogenetics component. But for this talk, I'm just going to talk a little bit about the macroevolution, specifically give you an idea of the regional diversity for the Caribbean in this genus, the timing of colonization, which means when it's estimated that the genus made it to the islands, and where it came from, the geographic origin. For this work, I did sampling all over the place. So there were more than 100 collecting sites, more than almost 600 samples collected, and they were scattered all over the Greater Antilles. I visited all the islands, mostly high elevation sites, and I also visited Guadeloupe, Dominica, and Martinique in the Lesser Antilles. To collect samples for the data I wanted to analyze. And I'm not going to get into details of the data, but know that I extracted DNA, generated a lot of data for different molecular markers. I used locality data to get an idea of species distribution. And actually, I should say that this was done in a global context as I incorporated data of Sticta into my phylogenies from other places in the planet. Okay. I also generated environmental data, mostly derived from remote sensing data. And I also calculated inter-island distances. And those last two components, I'm not going to get into a lot because that was used more for the community phylogenetics angle of the paper. But just wanted to let you know that this is a really comprehensive data sets that I analyzed for this study. Here are some cool views of these high elevation areas in these different islands. Here you're seeing Pico Duarte in the Dominican Republic, the Turquino, which is the highest elevation point in Cuba, and then the Blue Mountains Peak in Jamaica that I visited for the sampling in this study. So what I found many things, and I found that an estimated number of species for this region, potentially 64 candidate Sticta species for islands in the Caribbean. From my phylogenetic analysis, I estimate that around 60% of these are potentially endemic to the islands in this region. This is the most complex figure probably in the talk, but this is a phylogenetic tree, right? And I know most of you familiarized with it, but I should say, if you haven't seen them, this just traces and try to make the simplification of the way evolution proceeded and diversification worked. So we have each of the branches correspond to a species, a taxon, correct? And each of the intersections correspond to a node. That those nodes that right now have pie charts of different colors correspond to ancestor in the evolutionary history of the species in this group. So the thick branches mean that these are statistically significant, like strongly supported branches. The pie charts actually are the result of an analysis called ancestral range reconstruction, and the idea is providing an idea of the potential geographic origin of the ancestor that is depicted in that node, right? So if you see in blue, it says South America. That means, I don't know if you're seeing my pointer, but those blue pie chart mean that the ancestor probably came from and was only found in South America, correct? And the only thing that I want to say about this is that this is a dated phylogeny. That means that it's calibrated in time. So as you see in the bottom, we have time as an axis. The left part means that this is more remote in time, and to the right, it means that those events, those splitting events in the tree, happened more recently in geologic history. Okay, so what are hopefully, I hope I made a easy to follow explanation of this. But anyway, I just want to get into the main messages. First is noting that root node there means that Caribbean species, which are here mostly in purple, we confirmed something that I suspected from that Puerto Rican paper, that most of the lineages or the Caribbean lineages most likely came from early South American Sticta lineages. Okay, and that's that node represented by A. That Sticta arrived to the Caribbean potentially 19 million years ago. That's around the Miocene. And I also was able in this phylogeny to document instances of reverse colonization. That means that we could detect species that originally came from the continent, evolved in the Caribbean, and actually made their way back to the continent. As you can see, for example, in C, we have appointed here in C lineage that is potentially endemic to the Caribbean that eventually led to a node that is in green. And that green means in this tree that the species, the ancestor was probably distributed in the Caribbean and also Central America. So that's what I mean by reverse colonization, species that actually go back to the continent after actually evolving originally from the continent. So anyway, just take home messages about this study is that I think it makes clear that we have underestimated diversity and endemism in this region. This TICTA case made, we confirm strong affinities of South and of the Caribbean biota of lichens to South and Central America. Also, the case of reverse colonization argues against the idea that islands are kind of a dead end in the colonization road for species. And we can talk more about that, but it actually argues that lichens have the ability of actually dispersing back to the continent. So that's the first part of the talk. I wanted to quickly go over this population level study that I did with the genus that Natalie was talking about, Cladonia. This is a really cool study that actually started almost 15 years ago, and it entails the species Cladonia sanstidia in Puerto Rico. I've collected this for ages in the island, and as you can see in the picture, it's known from the populations are concentrated in two areas. One is close to Arecibo here in yellow. This is a coastal, hot and humid, low shrubland environment. And then the other population that we know of is high elevation, humid ecosystems, mountainous ecosystems in Maricao. This always blew my mind because they are considered the same species, right? These populations, but for me, the environments were so different that I was always suspecting there was something going on here, and maybe some subspecies. That this may actually be two different taxa. That's what I mean. So I did a quick pilot study, generated data for molecular markers, and I actually got different stories depending on the marker that I use. Here you're seeing EF1 and RPV2. EF1 saying that there's basically no differences between the populations in Vega Baja and Maricao, and RPV2 actually saying with strong statistical support that these are separate clades. The other cool and interesting thing I found in this preliminary study is that Cladonia sansterii was always found nested within Cladonia subtenuis. This is a more widely distributed species, morphologically really similar. So I found that fascinating. I wanted to understand what was the relationship, trying to disentangle what was going on with this group. So for that, I should say before going on, I wanted to talk more about subtenues, which you probably have seen in the areas that you live. But the main thing here is that they differ. Based on what was the species concept that was traditionally used. Subtenuis was more widely distributed in Eastern North America and the Caribbean, and Sansterii was mostly like a Caribbean endemic with populations known from the southern easternmost part of the U.S. And how lichenologists, what they used to separate these two really similar species was their chemistry. So Sansterii had atranorin and subtenuis has usnic acid. So that was the species concept was based on mostly chemistry, and secondary chemistry, I should say. So for this work, I went to all those populations, I collected samples, and that includes the easternmost part of the U.S., and also the islands of Cuba, Jamaica, and the two populations in Puerto Rico. I identified the samples, based on the traditional idea that was like really minor morphological differences and the chemistry to separate the samples with atranorin and usnic. I used to get the resolution that I needed, I had to use more like a different approach for the genetic data. So I used RATSIC, which more or less is a population genetic approach that samples the genome of lichens and get snippets of genes all over the place, and allows me to analyze all that amazing sampling of the genome at the same time with this software called Stacks Pirate, for example. And then for the analysis, I combine phylogenetics and also some population structure approaches that I'm not going to cover, but that I wanted you guys to know about. So here are my results. This is the phylogeny, similar, in terms of appearance to the previous one that we see, but several things to note here. So each branch again represents here individual, correct? And so that's the tree to the right, to the left, and then we have like a color-coded columns. So the orange column rows mean that sample we found atranorin, and the more greenish looking column means that sample we found usnic acid. And then to the far right of the phylogenetic tree, I coded the samples by geography. So in gray, we have samples deriving from America, from North America, and then the samples that I collected from the Caribbean, specifically the two populations in Puerto Rico, Cuba, and Jamaica. So as you can see, and I'm not gonna go into a lot of details, but what I wanted to point out here is that the traditional concept that was based on the chemistry is actually not valid, right? Because let's say that if it was valid, what you would expect is for the samples of Sansteria that has atranorin to cluster in a single clade, and then the subtenuous, most of the subtenuous samples that have usnic to cluster in a separate clade with strong support. And as you can see here in the samples from the United States, in this case, we have a mix of samples that have different chemistries. Actually, some individuals have both substances, some individuals had none of the substances. So there's no apparent correlation of chemistry with the phylogenetic structure that we obtained from this analysis. So what that meant is that we needed to challenge the traditional concept and come up with a new one. So what was our solution to this problem? Was that we decided to go with a subspecies approach. We left everything within the name Cladonia sansteriae, and the different populations based on their different geographies were assigned as different subspecies. So we assigned the subspecies subtenuis to the populations of individuals in North America. We decided to name the subspecies Landroniana, as you would expect, dedicated to Ismael Landron that I mentioned at the beginning, for the two populations in Puerto Rico. Cuba has its own subspecies, and that's supported, as you can see, the lower part of this tree, the thick branches again have strong statistical support. So we're confident that these are really kind of real clades in that sense, and that's the basis of all these designations. And then Jamaica, which is also a strongly supported clade, we decided, it's where the type specimen came from. So this correspond to the true and real subspecies Sansteriae. So that was our solution to this interesting case. So before ending, I think I have hopefully some more, like couple of minutes. You can feel free to stop to tell me that I'm getting short on time. But before going further, I just wanted to, as I mentioned in the short abstract that I sent, wanted to talk a little bit about challenges for Caribbean lichenology. I think it's important, right, to highlight. And these are some of the stuff that I'm going to mention applies to all organisms. Some are more relevant to fungi and lichens, and some are actually more pertinent to lichens. And this that I'm going to mention applies to everything, right? Diverse areas in the Caribbean are frequently the most inaccessible to get. And this is mostly due to the rough topography and really tough weather that we have here. And also something not mentioned much, especially when you try to collect in these different islands that have different government systems, different languages, it's really complicated to get your permits and everything. So I wanted to highlight that because this is an actual impediment to know more. I don't know if you're seeing this video. So just wanted to bring it to give you an idea of the difficulty of sampling. So this is an area that I went with my colleagues in the lower westernmost part of Dominican Republic, the Sierra de Bauruco. Really, really hard to access, and as you can see, this really wet all over the place, really hard to walk and really hard to access the most pristine areas of these ecosystems. So anyway, just wanted to show you that to highlight that first important point. Really hard to sample the region overall. I want to mention an overall lack of interest and public awareness, and there are several angles to this, and that happens also to many fungal groups, and not all of them. You know, we know that many fungal groups are like really the public tends to be attracted for different reasons, but for lichens, especially in the tropics where we have a vast majority of them are crustose, which means that they're not very attractive or charismatic for the general public. We have issues of having not many people interested in studying them and looking them. In Puerto Rico, I have had many students coming to me to saying that they want to do projects, and actually they don't follow. You know, it's a really steep learning curve with them. And high diversity, astounding diversity. So, you know, usually I don't get people that stay to continue the work. This image that I'm showing here is a nice example of the importance of education. This is a post, a social media post in the really well-known Puerto Rican group called Biodiversidad de Puerto Rico, where people post pictures about biodiversity and organisms in Puerto Rico. And this translates to, this is what's taken in Vieques, which is an island here. And it says, how can I control these white spots? Those white spots are lichens, right? So this person actually don't know that these are organisms that are not harming the tree. So again, highlight the importance of education and having more lichenologists doing the groundwork that is needed. There's also, I think there's a limited but growing use and a limited accessibility to citizen science platforms, especially iNaturalist, which is the most popular right now. And to illustrate that, I went yesterday, I extracted data for Lecanoromycetes in iNaturalist, and I extracted 41,000 observations for the state of Florida. And as you can see, there are observations all over the place in Florida. When you look at the really highly diverse, by far more diverse than Florida, Insular Caribbean region, we have really sparse observations, most of them from Puerto Rico, where I am at, correct? And my colleagues are also, you know, that know me are taking pictures mostly to send them to me. But again, you know, 3,000 observations against 41,000, it really makes, you know, I think drives the point that not many people are using these tools for different reasons, education, but also access to internet, for example, is limited. We have a lot of electricity. You have, how you say, like when the electricity goes out, correct? So those are actual limitations to know more about these organisms here. Talking a little bit about iNaturalist citizen science platforms, I want to bring the point of how these tools can also be a double-edged sword. I bet there are studies about this with other fungal, taxa, but a couple of studies are highlighting how tools like iNaturalist can be confusing, especially if you try to use the data from iNaturalist for like more formal studies and inventories and taxonomic work. So in this study from Monsi in 2023, she makes the case that, you know, a lot of this data should be used, but with a lot of caution, because we don't have enough people curating the data in this platform. So it's important to be used, but just to be used with care. Some of the issues highlighted in this study, for example, is that for lichens, as as Natalie perfectly illustrated with cladonia. It's just so hard to put a name when most of the stuff you need to see in the microscope. It's impossible. That happens with other groups of fungi, correct? But with lichens, we have really, really small lichens, especially here in the tropics. The lack of enough experts with a trained eye. So that means we're losing a lot of the diversity because you're not getting the zero point 0.1 millimeter apothecia that is really hard to see unless you have a more trained eye. Also, the issue about research grade misidentifications. As you know, with iNaturalist, it's not that hard to get a research grade identification. You just need two or more people to agree an identification. So especially if in my case in Puerto Rico, people know that I'm kind of an expert of this, and if I say like this is something, they agree with me, and then they raise that to a research grade ID. And if I got it wrong, then you're just identifying something wrong. Also, the invisibility I wanted to make about to mention invisibility of records and how that translates into unaccounted diversity. So many people, since they don't know what they're taking pictures of, they leave the picture without a tentative identification. And that's stuff that I'm missing because I don't get when I look at iNaturalist, I'm not able to capture and put a name into it. So again, always to the minimum say that these are fungi, including lichens category, so people like me and other colleagues can take a look at. I think I'm close to the end, but also wanted to highlight the issue of limited funding and scarce opportunities for research and academic development. This happens all over the place, right? That this is not exclusive to me and Puerto Rico, but it's especially relevant because I'm in a biodiversity hotspot, and that I know of. In the lichenologists with training in my region are just two. It's just me right now, and Dania in Cuba. So that's very little people looking at highly biodiverse region. And the reason is that, this that I'm mentioning, the limited funding and the limited opportunities, there have been job opportunities for academic positions here, but they rarely come, in focusing on people interested in systematics, taxonomy, and fungi in particular. So I want to end up my talk, hopefully leaving some room for questions with a conservation concern. So why is this all relevant? Is that we're losing in the tropics a lot of species. So what you're seeing here is an image, a satellite image of the how you say, like the frontera, the boundary between Haiti and Dominican Republic, and the yellow pins are the sites that I visited and that I show you in that video, where I collected samples of sticta for the first project that I talked to you guys about. And as you can see, this is agriculture moving north up the slope in high elevation areas in the Sierra de Bauruco, and I know of some of these sites that are already completely destroyed, and that my samples are probably the ones left from anyone that collected anything in this area. So that's I guess I wanted to end up with that, just to give you a broader perspective of the importance of having more work, not only with lichens, but especially less charismatic organisms like lichens, insects, fungi, but different groups of fungi that also need to be, and that actually, ironically, are the most diverse compared to birds, plants, amphibians, etc. We have more diversity in these groups. So I'm gonna jump that slide just to talk a little bit, to say thanks. I wanna thank you all guys for hearing me. My Ecological Association of Washington, to Tom, to Serenella, your program chair. Thank you, Serenella, for inviting me to this talk. It's always good to talk, and especially with a group like this. I wanna highlight the advice, friendship of my colleagues, Dr. Robert Looking of the Berlin Botanical Garden, and Dr. Thorsten Lunch, Vice President of the Field Museum, for their advice, support, and friendship. Funding sources, I got a lot of funding for the work that I show you from the University of Chicago, the Field Museum, and the Ford Foundation. And last but not least, I want to say thanks to my colleagues in the region, friends from all over the place in Cuba, Dominican Republic, Martinique, Jamaica, in Georgia and Florida, and here in Puerto Rico, Dr. Eugenio Santiago. To all of them, none of the stuff that I have been talking about would have been possible. So with that, I want to say thank you. And if there's time, I'm not sure. Serena told me it was like an hour. I think I'm hitting the 46-minute mark. If there's room, I'm happy to answer questions.
Of
Thanks.
course, there's room. And thank
Yes.
you for a wonderful talk.
Of course. Of course. Let
Thank
me
you.
stop
Yeah,
the
thank you so much,
share.
Joe. We do have a number of questions. I'm very
Awesome.
enthusiastic. We got a lot of feedback during your talk in the chat. So this is what I propose. I'll go through and pose some of these questions to you that were in the chat, and then I'll just allow everybody to come off mute once we work through these, and we can hang out as long as you're willing and able.
Definitely. And I actually, if I may, I wanna, it might sound like a litany of apology, but I know I went fast over some of the phylogenetic trees and the details, but I was, I know this is a group that I can easily, keep up and, see what I'm trying to say, and you can read all those graphs. So I'm sorry if I went too fast in some of them, but again, the question session is for that, right? Like I can jump back some of the slides and talk in more depth about it.
I didn't find you went too fast. My only follow up is I think you and Serenella need to
Yeah.
organize a trip for the club to Puerto Rico, so we can put some of this new knowledge to into the field.
Don't tempt me, Mr. President.
So we got a number of good questions in
Should
the
I?
chat box, and I'll start with John Butler. He asked a question at the beginning asking about whether or not lichens grow inside of caves.
Good question. So we have, thanks for the question. We have a lot of caves here in Puerto Rico. We have hundreds, if not thousands of caves. Most of them we know where they are, but we're still discovering new caves all over, especially in the northern karst region of Puerto Rico. So inside caves, inside where it gets really dark, we don't get them, right? Like it's just hard for the algae or the cyanobacteria, especially, they need that light to survive, and the lichen too. But I have found really interesting stuff growing, of course, in the entrances and getting really far inside the cave where there's very little sunlight. But those the issue with rock dwelling lichens, as you can imagine, is collecting them. It's just hard to hit the rock and grab a sample when you're doing like a really long hike and having to grab rocks in your backpack. And I love rock dwelling lichens, just a pain to sample them, and they're really small. It's just a mess. There we don't have enough. I don't think we have enough um, keys, I guess, taxonomic keys to identify a lot of those. Depends on the group that we're talking about. But we do, so to summarize, we do have them associated with caves. I don't know if there are lichens that are specialized in the cave environment, right, that have evolved in cave or close to the entrance of caves that are strict from those environments. Don't doubt that there are, but I haven't looked at inside the cave. Definitely, I haven't been able to see anything really.
Okay. Cindy asked a question before. I'm going to kind of paraphrase. She was basically asking about the species diversity that you find in the Caribbean.
Right.
Is there a island effect to this? Kind of like what I think of as with the beak of the finch argument with Darwin and the Galapagos, where you find that a lot of these lichen species are endemic to just one particular island, or is there
Right.
a lot of overlap across the Caribbean?
Awesome. And just saying that, I realized that I didn't, mention a lot about the one of really key points that I wanted to make is the underestimated endemism that we think it's happening with lichens, and that it's remaining to be uncovered. Um, yes, as I said, for example, for the STICTA study, we estimate close to 60% of those uh, species to be endemic to the island. But that's, of course, we have to say that that's a tentative conclusion, because since I have already mentioned that there's not many people looking at lichens, and there's not many lichenologists overall in the planet. So it's hard to make the case that anything is endemic. It's just that I randomly collected it, and since I'm the only guy looking at them, it makes sense to make the call, that this is a preliminary or a tentative endemic, but that's pending additional data, additional sampling. I wanted to make a quick note, because you said something about Darwin and the beak of the finch, right? And that makes me think a lot about the interesting topic of unique, let's say, like morphological or anatomical characteristics that could be related to evolving in an island environment. Um, in lichens, I'm gonna give an example. One of the two genera that we described in that 2014 paper, in the family Graphidaceae, one of the genera that we described had something that was not had some morphological characteristics that were not known from other members in that family. So to the degree of how that is related to evolving in an insular environment, what's it, if there's like a direct relationship there, I'm not sure. But I think overall the set of environments, the different ecological aspects that probably are playing, like for competition for resources, competition for space and food, etc., are probably driving uh, uh, diversity and evolution. That's crazy in this island. It's just that not many people looking at this and quantifying, the degree to which we evolution have played out in this area. So the question about endemism, I focus in my work mostly on sticta, uh, but work by Robert Looking and some other colleagues that are working on specific islands have also uncovered higher uh, potential endemism in other genera. For example, that Taxilova um, genus that I mentioned when I was talking about Robert, that's only known, that genus is so far only known, if I remember correctly. There might be some recent paper here and there, but um, only known from Hawaii and Cuba. And those are two island ecosystems. So again, anyway, there's a lot of room to talk about this, but yeah, thanks for that question.
So, John Butler asked another question
Yeah.
regarding migration of lichens from one island to another in the Caribbean. And, typically, whenever you think of the Caribbean, you have the tropical storms that are whipping up all the spores, and I'm talking about all kinds of macrofungi now, and broadcasting that over a far greater, larger area. Of course, with lichens, the working presumption is a lot of their reproduction is done asexually, say through fragmentation, whatnot. Any thoughts on how lichens are moving around from island to island in the Caribbean?
Yes. Thanks for that question. I mean, I have thoughts about this. You know, the presumption with lichens, as many fungal groups, is that they're easily dispersed, correct? Because we are talking about spores or really small um, fragments of lichens that probably can be picked up easily by wind, and even organisms and animals. all around. So, but again, you can be really small and probably you you probably can have the morphology that allows you, that puts you in a good position to be moved from very to very distant places easily. But one thing is that, another thing is the different factors that happen during the migration, right? Like the spore or the fragment needs to survive a really harsh environment, which is the atmosphere, right? That is exposed to UV and stuff like that. And you need to assume that you will find a suitable habitat to grow, and that in the case of spores, you have to assume that you will find a suitable photobiont or algae or cyanobacteria that is compatible with you. So again, many factors that are controlling this. About this, I didn't mention it because this touches more on that component of community phylogenetics that I omitted for this talk. But one of the things that I mentioned, so something I found with Sticta in the Caribbean, and that still needs a little bit more data, is that Puerto Rico and the Lesser Antilles, their communities had a tendency to have uh, more shared taxonomic and phylogenetic history. So there was more connection between Puerto Rico and the Lesser Antilles, and then the communities in Jamaica and Hispaniola were more closely related to each other and had closer affinities. So one of the arguments I make in that paper is that if you look, it's hurricanes actually. This is a active area of hurricanes. And if you look at historic data of paths of hurricanes, you will see that there's a slight tendency of hurricanes to tend to connect more the Lesser Antilles to Puerto Rico. So when we have an eastern, a hurricane that is going to hit us from mostly central part to the east, those tend to have crossed already some of the Lesser Antilles, and then they continue their way up. So that was an argument I made in that point, because honestly, I cannot think of anything else right now. Besides that, I can envision small pieces of lichens probably flying in these hurricanes and landing in Puerto Rico, and probably that serving as an explanation to similarities between those communities. But this is, a fertile earth and terrain to explore. I don't know how to say it, because no one is looking at this, honestly. I just make inferences about what's probably happening, but I don't have the data to support it. But yeah, that's an excellent question.
Yeah, yeah. If there's one overlying theme in tonight's talk, we need more eyes out in the field. I'm going to jump a couple questions down because to my mind, it's a little bit of a philosophical thing, and I'd like to hear your thoughts on it. So Stephen Buckland asked, "What the heck is going on in Cladonia? Were there any morphological differences or differences in photobiont communities between the different subspecies?" And it makes him wonder, and me wonder as well, how many other traditional species concepts in the genus are going to stand up to thorough genetic scrutiny.
Yeah. So what's going on in Cladonia? I want to make the note that I don't think I'm an expert in Cladonia. And Cladonia is a really special genus of lichens. So really diverse and very similar looking. It's just a mess depending on where you're landing at with this group. But yeah. So you mentioned photobionts. I think there's something potentially happening with photobionts, and when I was analyzing this data, so the good thing about RADseq and the approach that I used for analyzing this data is that I not only obtained data from the fungal counterpart, but I also obtained data for the photobiont. I think it's Asterochlorus, the genus that occurs in Caledonia, but there's maybe someone can correct me here. I don't remember. But I do remember that I tried to filter my data and try to assemble a data set of the photobiome components in my populations. And I did get some structure that, if I remember correctly, was separating North America from the islands. I don't remember the details, but I do remember I got a pattern. I am this I need to talk more about this with my colleagues in the Field Museum, especially Thorsten and some of the people more expert in rat-seq and population level analysis. But I think photobiomes are definitely something to look at to understand what's going on with Cladonia subtenuis and Sansteriae. But yeah, that's what I can think about to mention about this. What else? What else? What else? I mean, for the continental populations, it didn't really surprise me a lot. What I found, especially in North America, as you saw, that's a that was an assortment of species of individuals with different chemical compositions. Some of them closely related to. But I mean, when you look at the tree, that side of the tree has no clear pattern, which tells me that these individuals are probably easily dispersed in this continuous environment that is the land in the continent. And there's like a sharp contrast when you have put water in between, right? And that challenges a lot, our notion of these individuals as easily dispersed and having really, how you say, like broad distributions. I don't know if I answered the question, but anyway, I want to look at that more closely.
When you did delve into some of the chemistry associated with lichens, they're chemical warriors, which brings me back to another earlier question. Do lichens have any traditional usage for, especially like amongst the native peoples in the Caribbean? I mean, you always think of lichens as being used for fabric dyes.
Correct.
Anything come to mind?
Unfortunately, I'm not sure. Like, I don't know. Like, no one that's angle of a lichenology, right? And that links the people to the organisms. There's not many. There's basically nothing. I do remember. The only thing that I can remember in my years working with this is there's a person here, also like an amateur botanist, but also specializes in using plants for medicinal purposes and stuff like that. She once mentioned that she did. I mean, this is a person that did interviews to our elders in the different areas in the island, and she was trying to compile, traditional botanical knowledge from the different elders and communities in Puerto Rico. And she mentioned me the use of usnea, which is the bearded lichen, for teas and how you say teas, like infusions. I don't know if I'm using the word correctly, but she documented people mentioning using usnea for treating stomach ache and like not liver, but how you say riñón, kidney issues. So, but again, she didn't mention much more. I don't know from where she obtained this information. For the rest of the islands in the Caribbean, I wouldn't be surprised. I just haven't looked at it. For some reason, I would not be surprised that Cuba, you would find much more information. Of course, there's more people there, and it's a much bigger island, so there's more species, so there's more probability of people potentially using this organisms. But unfortunately, compared to what we know, like the example you said about fabrics, and there's a lot of documentation of lichen usage by traditional peoples in Mexico, Central America, Peru, etc. We don't have a comparison. Like it's not comparable because no one has documented any of this, unfortunately. But I don't doubt it that our native indigenous, like the Tainos, for example, probably use it. It's just not documented that I know of.
We had another question about the bedrock geology and how it affects species diversity. And I think I know at least part of the answer. In the Caribbean, I would imagine that you have a lot of mafic rock, basalt types of rock, and a lot of calcareous rock, corals and limestones, etc. And of course, lichens care a lot about what they grow on. So
Right.
especially for the lichens that are saxicolous, that are growing directly on the rock, you're going to have different lichen species showing up, just completely dependent on bedrock geology. Is there anything more that you can expand on that?
That's a tough one. I think the what I can say about that, I think that first of all, that I don't think we would need first to look at saxicolous lichens. That would be a first place to look at, like just starting to look in more detail at this species and what could be going on in terms of the trying to reconstruct their do phylogenies and try to correlate their specific geological substrate with what might be going on in their um, evolutionary history and the patterns we get in the phylogenies. But of course, when I think of this, I just think of indirect effects, right? Like the Cladonia case, for example. The populations in Puerto Rico of this Cladonia sansteriae that I mentioned. I said that these are from two different contrasting environments, and the main thing that I think, besides the weather, right, the temperature and rain, that separates these two areas is their geology. Like one is the one in the north, in the coast. These are siliceous sand, like white sand environment. And then the Maricao population, this is the high elevation population. This is a serpentinite, or I don't know, I think it's serpentine or serpentinite derived rock. And the trees and the plants that you get there are really special just because of the geology. So when I say indirect effects, is that I think that whatever we're seeing is through the lens of those substrates where the lichens are growing, which in this case are trees. And these are special trees that are growing there because of the special geology. So that's the way I kind of connect what's going on with the tree dwelling lichen, what might be going on with the different geological substrates. But to make a direct um, comparison or link is way harder, I would say. But I don't think there's anything wrong with my approach. How I'm looking at it. It's just key and always important to mention that this is indirect, like an indirect connection that we are trying to make. Like but yeah, unless looking at saxicolous lichens or species that are more uh, growing and dwelling in soil environments or rock environments, it's just way harder to make stronger connections. I don't know if that answered the question, but yeah, that's what I would
Yeah.
say.
Yeah. I don't remember
Great.
what slide this was asked on, but Camille227 asked if the Maricao and Vega Baja are the same substance.
Species. So Camille 2277 is Kurt Miller. So hi Kurt, thanks for the question. So yes, the Maricao and Vega Baja, from what we decided to do with the Cladonia sansteriae mess, we decided to put both populations under the name, under the subspecies Landroniana. Um, so it's Cladonia sansteriae. So the species doesn't change for Puerto Rico, but now it's its own subspecies, which highlights, its uniqueness in the context of the rest of the Caribbean islands. However, I should say when you look at the phylogenetic tree that I showed you, there is strong structure in those clades, with the minor issue that the Maricao clade is paraphyletic. So we have a few individuals that get to be closer to the, phylogenetically to the Vega Baja population. So that kind of made me, hold back and play it safe and say, let's just put everything in Puerto Rico under the same subspecies pending of more data or more detailed analysis. But I wouldn't be surprised that a more extensive genomic sampling of this population would reveal that these are actually separate, or at least it depends on who you're talking to, right? Like naming a species or a subspecies, as you know, could be really subjective depending on the particular group of people looking at. But I wouldn't be surprised that some other uh, groups of researchers look into more detail into this, and decide that these are actually two separate species. But honestly, yes, Kurt, these are. We decided to just name it all Landroniana, to play it safe until we had more data. Yeah.
Okay, I think that's pretty much the end of the questions in the chat. Chris,
Awesome.
thanks for being so patient. You've had your hand up for a long time. You can, yeah, I see you just came off mute. Okay.
Yeah, thanks, Tom. The question I had, and I admit it's a little off topic, but first off, let me say I really enjoyed the presentation. It
Thank you.
was immensely detailed, and I applaud the amount of work you've done. My question goes back to something that I experienced as an undergraduate back when the Earth was still cooling back in the 70s. And the professor I worked for, invertebrate zoologist, fortunately had a daughter who lived in the panhandle of Alaska. She would send us bundles of lichens every few weeks, and we would put them in a Bierman funnel and extract nematodes and tardigrades and ciliates and flagellates and all kinds, and probably some things we never did identify. And I found it absolutely fascinating because on a couple of occasions, her samples showed nothing. And we said, well, what happened? And then she dug and found out, well, the U.S. Forest Service was aerially spraying herbicides in the area that day. Now, externally, the lichens looked fine. They looked okay. But their contents were next to zero. And so I had sort of like two questions. Number one, do you know of anyone carrying on that kind of research? And number two, does anybody have any idea what all these, we refer to them as cryptozootics? Does anybody know what any of these are doing inside of lichens or even on the surface of lichens?
Well, yeah. So I think I might have to ask you to repeat your first angle to this question, because I wasn't sure if you're talking about, like, for example, the part that you mentioned, the herbicide experiments. So you're talking mostly about work done with cryptozootics in lichens overall. Like, what do I know about those type of studies with, like, say, lichens in the Caribbean? What was that? Am I
Or
getting that
anywhere.
right?
I am
And the
actually
role
getting
that they
back
play?
into it myself now. I mean, I've collected some lichens. I've got a scope. I'm starting to do some of my own studies locally here. But what
Yeah.
fascinated me was years later, after this episode, I was judging a science fair, and the two brothers who won the science fair did their project on what they called cryptozootics in lichens.
Uh-huh. Uh-huh.
I thought, well, wow, we weren't the only ones looking into this. And I thought it was absolutely fascinating because throughout my biological career, I mean, it's all about what else is growing with something else. Nothing
Right.
lives alone. And
Correct.
so I try to, fathom what does that tardigrade or that talina, that ciliate have to do with this lichen? Is it just along for the ride, or is there something going on here we should look into? So I just, thought, well, if anybody would know if this research is still going on, you might be aware.
Yeah. Well, yeah, I got you. Well, first I should say that I said it previously before the start of the of this talk and this meeting. You probably heard from my introduction that I still do lichen work, but I had to do other work to pay my living. So I'm doing more like independent contractor stuff. So I should make that distinction because unfortunately, I'm not that active into research in lichenology. I still do my stuff. It's not as intense and active as I was just a few years ago. Just making that note there. But having said that, so that's really interesting overall, and I think that's really one of the next frontiers with lichens and probably other groups of fungi, right? Like, I don't think there's many people looking at this. I have encountered these microorganisms when I make my preparations of when I'm looking at lichens, and I see them, but I always wonder what's going on with these guys. I have no idea what they're doing. I wonder if they're specific to some species or some group of lichens, if there's specificity, right? Like really strong links between the communities in both groups of organisms. But that I know of, at least in Puerto Rico where I'm living, I don't think there's anyone looking at this. The issue of the potential effects of the use of herbicides and stuff like this, and how that can affect populations of these organisms living within the lichen community environment. No idea.
Yeah, I guess the other thought I had was
Yeah, it's
just looking at
fascinating,
air
right?
pollutants.
Yeah,
Where I live, we've seen severe ozone damage on garden crops, believe it or not, in our urban environment, despite the lack of major manufacturing. And so who knows? I mean, I look at it as a canary in the coal mine. Maybe these
right,
lichens have the ability to detect issues
right.
before we do.
They actually do, right? Like probably how I got into lichens was of what you're saying. I did my undergrad thesis looking at the effect of sulfur dioxide and the communities of lichens nearby factory, a glass factory here in Puerto Rico. Of course, I was just an undergrad. I didn't have enough resources to look at this in like a lot of details. I did detect changes in the diversity, especially the species richness, I should say. They tend to have a relationship with distance, right? Like more species of foliose lichens more distant to this factory. But besides my work that I did as an undergrad, I don't know of any other that has been done, but the relationship between pollutants and lichen community diversity and composition is really well established. The pioneer work has been done mostly in Europe and in England, but there's many studies of that in North America, Canada. But unfortunately, in the neotropics, especially in the Caribbean, close to nothing. And one of the main issues, as you can imagine, is that this is a diverse atmospheric environment. We have northerly, easterly, how you call them, the winds, or easterly winds that come and makes our atmosphere really dynamic. So we have a lot of flux of air currents that makes mixing of all these pollutants, and they are carried away really fast and easily. And it's really different to what you see in continental areas, right, where the smog and other pollutants tend to stay because the atmosphere is more static in that sense. So here it's way more challenging. It's not. I'm not gonna say that it's not happening, because we do have pockets of atmospheric stability in the Caribbean and other neotropical areas. It's just that there's not enough people looking at it, and I'm not gonna say that it's because you know there's a relationship that people in South Central America and the Caribbean less interested in lichens. That also there's limitations in terms of funding, in terms of resources,
Of
um,
course,
which
yes.
makes more relevant the importance of this type of talks where I can connect with all of you guys, right, and exchange ideas and all this idea of networking, um, to help each other, right? Like by connecting with you, I know that I can probably expand, hopefully at someday, the reach of the work I'm trying to do. But anyway, I just
Well,
also
thank
thinking of
you. And if I
one of
find anything, I'll write you.
definitely. Thanks, Chris.
Okay. Does anybody else have any other questions before we finish up? It's just about at the 9 o'clock hour. Joel, muchas gracias. That was outstanding tonight. I so enjoyed that. Very
That's
good.
amazing. Thank you.
And with your repeated calls for more eyes in the field and all the direct flights to between DC and Puerto Rico, I think we need to get working on having a trip for the DC club in the upcoming year. Wonderful.
Hope so.
And
Yeah.
and
Happy to have you guys here. Let us know, and we'll go do field trips and stuff.
Okay. Good night, everybody. Good meeting. Thank you. Okay. See you in person next month. All right. Good night, everybody.
Bye.
Goodbye. Thank you.
Adiós, Joel.
Adiós, Serenidad. Hablamos luego.
Hablamos. Chao. Thanks for hosting, Tom.